Hearing my voice is really a wreck. Today I want to talk about how I've used ISMB in biochemistry and trying to integrate it into other disciplines as well. All right, going this way. I've done modeling since 1985, and we've heard a lot of talks about the progression over time as we've gone from Silicon Graphics workstations down to web-based programs. I didn't know about iCn3D until around 2016, 2017. I was at an experimental biology meeting, walking around to the NIH booth, and I was looking for a program because I've written an online biochemistry text, which I could plug in the actual molecular models. And when I found out about it, it was like a switch flipped in my mind, and I never went back. It's an amazing program. So today I want to talk about three things. You're not going to use it unless you know about its advantages, and I think those have been clearly articulated today, but I'll talk about what I think they are and how to integrate it into biochemistry courses and curriculum. But yeah, as a chemist and biochemist, there's a weak link to everything, and I think we need to start molecular modeling early, high school, and all introductory college courses, and at least in chemistry and biology. So I have some ideas about that as well. Okay, advantages. Everybody knows the advantages here. I've colored two. The saving rendered states is an amazing pedagogical tool. You can get students to send you their state. You can give them a state, and they can remodel it. Also, I have something here that's supposed to be a joke. When I went to the meeting in 2016, it said this was going to exist in perpetuity. And that was great because I switched so many times from different modeling softwares. But of course, we know that perpetuity means different things to different people. And of course, it's connected to a lot of databases as well. One of the disadvantages is it's not as pretty as Primal, but who needs pretty all the time? In fact, you need it very little of the time, especially as you age. So today I want to talk about how I've integrated it into my biochemistry courses and materials. I've written an online biochemistry text. It's a member of the Libra text out of UC Davis. It's called Fundamentals of Biochemistry, written by myself and Patricia Flatt. Now I'll refer to it as FOB. Here's a QR code, and I'll briefly explain a little bit about it. It's a 5-volume series. It is huge. I mean, I didn't plan it that way. It's probably about 2,500 pages if you print it off as a PDF. But it's based on the organization of almost all biochemistry textbooks. Volume 3 is information pathways, and I think the secret is that no one ever gets to those in a biochemistry course, but even in a 4-year sequence, but they're there anyway. Today I want to spend most of my time talking about, well, a little bit about part 4, but mostly part 5. I don't have questions, end of chapter questions embedded. Instead, I have a whole volume. 1 second, I'll get to that. That's volume 5. Volume 4, since I wrote this, I don't have to worry about what an editor says. So what I have are special topics, something on biosignaling, 240-page chapter, integration of metabolism, and something very special to my heart, biochemistry and climate change. It is where I'll spend most of my time today is looking at this, volume 5, that has literature-based guided assessments as well as ICN3D tutorials and help. All right. So I have approximately 600 ICN3Ds, pre-rendered states, percolated throughout the book in all these different areas. And of course, you would expect that if it truly covers a whole year course. I'm just going to illustrate 2 points here. Obviously, all these structures have textual information associated with them in the book. They don't stand alone. But the first one here is diffusion across the membrane, pores. So I have, for instance, a pi mole morph of a small conductance mechanosensitive protein. You can see it's changing its structure. But then in the same chapter section, I have plenty of ICN3Ds. This happens to be cytolysin A, a monomer. The cyan indicates hydrophobic areas. When you get a lot of these together near the membrane, they can self-aggregate through a conformational change shown here, and they form this structure. And this is what I have in the book. So for instance, you can click on that ICN3D model, and up pops a rotatable full menu ICN3D menu in which you can interrogate the molecule. There's also a short link, which everybody talks about. And if you go to that link, you get the exact rendering. This is simply a top-down view looking down the pore. And you can see there's a red and blue dummy atoms that represent the outer leaflet and inner leaflet. And when I first started doing this, there was no program that did this. Now I see that Molestar does it as well. This is chapter 28.5, small G proteins, gaps, and GEFs. I have a lot of images in the book that I did not create, but I have Creative Commons permissions to reuse them. So this is an example of KRAS, and it has motifs, and they're color-coded in a static image. I've been able to convert most of these to ISMB, and so you have a rotatable image of the molecule as well. Okay, now I've been teaching for 32 years, and I don't know if molecular visualization has helped students really understand structure function. So how do we know? So I'm a part of a group called BioMolViz, and what we've tried to do is develop and support biomolecular visualization literacy. And how do we do that? We devised a series of 12 themes, overarching themes, that we have to intentionally address when we teach structure-function relationships in the course. And I'll talk a little bit about these. So we've created a framework, and for every one of these themes, we have learning goals and objectives. So this is one example, alternative rendering. It's the theme. And so, you can present students with these, and who knows what they think or see when they look at that, but it's the same structure. But if you actually look at the learning goals associated with these, here's one: students can create meaningful molecular images, blah, blah, blah. And the next one, students can produce the best rendering of a macromolecule. So this calls for iCn3D use by students to actually create, not just look at rotating images, with the hopes that maybe that will help them develop the literacy that they really need. I'm just going to show you several examples. We've already seen a bunch of examples. For instance, for the theme alternative rendering, here's two examples. One, these are tanned repeats in annexin A5, and you can clearly see them rendered in different ways. This is the GP120 core, where there's a white surface, and the glycans are shown in both stick and cartoon fashion. So what you do is you pick an image, and then you can assess specifically at that image related to a particular theme. Here's another alternative rendering, and we've seen these before. This is superoxide dismutase. It's the dimer, homodimer. This is another one in which the Delphi surface potential is shown here, and in one of the monomers, and you can see the zinc right there, and the blue around it is positive potential, and you can imagine the superoxide just zipping in there to the active site zinc. I love membrane proteins. I worked with one for a long time, but this would fall potentially under any of the categories, but it depends on the question that you're going to ask students. So this is macromolecular assemblies. So I have a peripheral membrane protein here on this side, and over here I have a rhomboid protease. And again, you can see the dummy atoms representing the outer and inner leaflet. After teaching 32 years and then having to use my hands to show what a protein looks like, and then giving them this and wondering why they still don't get it, I think it's because we're not intentional. And I truly believe, I'm just politicizing here, that unless we intentionally address biomolecular visualization literacy, it doesn't matter if we use VR or AR, students still won't get it. We saw another example of this mutational analysis, but for instance, one of the themes is structure function, monomer building blocks, and molecular interactions. We've already seen one of these 2D interaction maps a little bit ago. This is one of my favorite ones. This is JAK kinase dimer bound to a cytokine receptor. That's obviously not a PDF structure, that's interferon. And for this particular structure, these are all in the book, with associated textual information. You could address the themes of topology and connectivity given the color coding these motifs and domain structures within a single protein, macromolecular assemblies, and also symmetry and asymmetry, which typically is a hand wave for courses. We just don't deal with it. Again, we've seen you can overlap or superimpose images, and this is a particular one in which we have AKT, which is protein kinase B, in the active and inactive form. You color code them with the end lobe, catalytic loop, and activation loops to see their differences. Structure function. And of course, I've populated this with AlphaFold. AlphaFold wasn't even available when I was starting to write the book. And the ICN3Ds that I made 6 years ago, I would probably change it now, given the new functionalities that they constantly had, which I think is wonderful. That keeps me on my toes. And I found this really interesting. Glucose 6-phosphatase, a really important protein in gluconeogenesis. As far as I know, there's no structure of it. This is a AlphaFold model of it. So now I want to go to volume 5, which is assessment. How do we know the students know anything? So I want to briefly talk about the literature-based guided assessments that I've created. What I've done is taken papers that have Creative Commons permissions, and I can use their figures, graphs, all kinds of data from images directly in the book, and then ask questions, iterative guided questions, so the students can read the paper and slowly understand it, or they could do this first and then read the paper. So I've written several of these, actually many of them, some that I call traditional, some that I call global challenges, because I think biochemistry needs to be relevant to the students. So for instance, some of the LGAs, guided assessments that I've written, one's on climate change, one's on trauma, one's on addiction, one is on data integrity, kind of things you don't usually find in a book, like I just said about biochemistry and climate change. So I feel really good about that. So I want to talk about the other part of volume 5. It has ICN3D tutorials, and there's two kinds. One is this one. It's basically tied to all the themes of BioMoViz. So these are the 12 themes. Notice this is under construction, and I'm constantly seeking collaborators to help me write. Hasn't worked so well. Partly the pandemic came in and no one had time, and partly no one has time anyway. I'm theoretically retired and I have time. So one of the themes is macromolecular building blocks. So what I plan to do is have a pre-made ICN3D for a protein, and then have questions. You build one now, the user, for RNA. You build one for DNA. You build one for a glycan, so they can start to differentiate the monomers in each of these polymers. We're getting there. Basic skills. I have a lot of basic skills put in here. I think what's really critical is that ICN3D has to be expanded to be used in high schools and undergraduate courses as well. If it's so easy to use, why is it not? And I think high school standards have been set up that you can easily show that ICN3D would be really important in them. Obviously in intro bio and intro chem, but there's a cultural divide between these two disciplines played out in real life and also within chemistry. It's so easy to use. You can use, for instance, PubChem images to actually display. So there's so many molecules that you can do an ICN3D that would fit in high school and college courses. What's not present is inorganic molecules, but this is an incredible website for Chem3D. And I have one more slide that shows specifically that if you go up to structure function, bio-inorganic chemistry, you get a JS mole, a rotating molecule, and a static molecule. And I think the people at ICN3D need to branch out so that they have this kind of tutorial that would be available targeting next-gen standards in high school and also introductory bio and chemistry courses. So that's it. Thank you.
Yeah, very nice, Henry. So time for a couple of questions.
Henry, first of all, a really nice talk. It seems like we could classify you as an ICN3D super user.
Yeah.
And you mentioned that you have so many examples and you've had like light bulb moments with ICN3D. So I wondered if you could talk briefly about an insight that you gained using ICN3D that maybe wasn't possible with a different molecular viewer.
Well, I think for me, it was from a pedagogical point of view. What can I do to get the students to get something? And, it's something as simple as displaying a membrane with dummy atoms, to me, was profound. And then also the ability to make multiple renderings, and especially when they added the Delphi potential, looking at electrostatic potentials, there are so many things that would be useful to actually get people to maybe have an aha moment. But for me, it was the fact that they could make shareable links, and then I could interrogate them, they could interrogate mine, and go from there. So
great. Thank you for the insight.
yeah. Okay.
I have a quick question, which I know the answer to, but
I probably-
I just want to vent.
Yeah.
As part of an example of decline in civilization, you no longer both see 3D models.
Mhm.
Nobody knows how to cross their eyes anymore to see in 3D. And I find it very, very frustrating. So Philippe assures me that the problem will be solved with virtual reality, but I just don't know how you look at a 2D picture and think, even if it's rotating, and think you see something. Yeah, but they don't even, in publications, they don't publish stereo pairs. If you just cross your eyes, you don't need glasses. It's a lost art. So just a comment.
Some of us can't because my eyes don't work that way, but I understand your point. I get your point.